I'm going to discuss the ASPECT trial, as you've heard about. I'm the chief investigator for a very large trial group of about 85 centres in the UK and one in Canada. And this was, first of all, started in 1999, so 19 years in evolution. Now, why were we put together by the Medical Research Council in the UK? It's just, these are data you're familiar with, which is that there was a very high incidence of esophageal cancer, particularly in the UK. In fact, there's nowhere in the world that's got more esophageal adenocarcinoma than my home city, Glasgow. In fact, the cancer prognosis of people there hasn't achieved much improvement over the last 30 or 40 years. We also know something about the pathophysiology and the pathology in particular. We know there's a pre-malignant lesion that's present at least in 60s, maybe as high as 70 or 80% of the cases, called Barrett's esophagus. And last of all, we understand the pathophysiology and the physiology in particular, that the more acid and indeed bile and inflammation you have, the more likely you are to be predisposed to getting the esophageal cancer. So we looked at an evidence base of 20 different chemotherapeutic regimes with way of chemoprevention, including surgery, and we chose two specific regimes. We chose high-dose proton pump inhibitor therapy, 40 milligrams of esomeprazole twice a day, and we chose a low-dose regime, which is 20 milligrams of esomeprazole. Originally, I should say, we did want a placebo group, but that was deemed by the funders and the regulators as being potentially unethical, because these patients would be allowed access to proton pump inhibitors, which we were quite comfortable with to change our trial design. We also used low-dose aspirin. We chose 300 milligrams, which is equivalent to 320 milligrams in the USA, versus no aspirin. So what could be simpler? Well, actually, it's much more complex than that, because if you move to the far side of the slide with the composite endpoint we chose, is if you look at all patients with Barrett's esophagus, in fact, only 2 or 3% of individuals have a lifetime risk of esophageal adenocarcinoma, so we would never have been able to accrue enough events. So what we wanted to do was to actually look at the death in these individuals too, so we chose a composite endpoint. And these people are at increased risk of death from other causes, which we can discuss later on. If you look at our power calculations, which we needed 196 events, in fact we achieved more than 50% more than that. We had over 300 events, so we were certainly statistically powered. And to move to the results, in fact, we showed that with high-dose proton pump inhibitors, there was a genuinely very strong, significant effect compared with the low-dose proton pump inhibitors. So in other words, the 40 milligrams of esomeprazole twice a day was much more important in preventing the prevention of the endpoints than the low dose. Aspirin on its own, compared to no aspirin, didn't seem to have any statistical effect. So these were the main endpoints, which to us were in some ways surprising. And when we look at the secondary endpoints, in fact, the proton pump inhibitor had the strongest effect in mortality analysis, and in the slides which are not for publication, which you've been given copies of, you can see that in fact most of the effect of proton pump inhibitor is working on the prevention of mortality. However, when we censored for the use of non-steroidal drugs, and this is very important, some non-steroidal drugs have very similar effects to low-dose aspirin. So when these patients started it, we're talking about a median of 9 years follow-up, still ongoing. Some of these patients would have indiscretions and protocol violations, and we censored the start of that drug. We found that aspirin then had a significant endpoint. So there could be some effects with contamination of non-steroidals, giving us a clear signal. What was also important too, that when we did some sensitivity analysis, now forget this is not powered, we're looking at each of the individual arms, so you have high dose, low dose, proton pump inhibitors, aspirin, no aspirin. So when you look at the arms of these studies together, in fact it was quite clear that the treatment that had the best effect was a high dose proton pump inhibitor combined with low dose aspirin. Perhaps to me, because I've been the biggest skeptic of this trial, in fact everybody in the trial team knows that, I've always been looking for flaws in our own data. The thing that caught us very well aware here too was in fact that the total number of serious adverse events was exceptionally low, and that's very important in chemoprevention, which is actually you've got to have a benefit and a very low risk ratio. So on that basis too, there seemed to be a benefit. Thank you. I just want to thank the trial patients, the people who sponsored it. In fact, and sadly, one of our investigators, one of the senior laureates, Doug Altman, died yesterday, so I just want to thank Doug. And I particularly want to thank our funders for sticking with us for 19 years.